Hey guys, my name is Nina Kumar and I'm from Nutrition course. And today, me and my group mates are going to explain you guys about hematology system. And for my part, I'm going to cover you guys about the composition of blood and also the structure and function of plasma. Once your blood makes it to a lab, the technicians put in a centrifuge which spins around fast enough to send the heavier components to the bottom of the tubes and bring the less dense elements to the top. As a result of centrifuge, the three distinct layer emerge. First of all, down of the bottom, we have got a heavy red layer of electrocytes, or also known as red blood cell, which is to carry oxygen and carbon dioxide. They make up about 45% of your total blood volume. You have got this thin little whitish layer in the middle. Those are your barriers. The leukocytes are also known as white blood cells that defend your body from toxins and foreign microbes. And there are also cell fragments called platelets, which help with blood clotting and make up less than 1% of your blood. And the third layer is plasma. When we focus to the plasma is 90% of water, but the other 10% is chock full of 100 different solutes, including proteins, electrocytes, gases, hormones, and base products. Most abundant of these solutes in plasma are electrocytes, which you may hear of the secret ingredient in sport drinks, but they have just positively charged like calcium, sodium, and potassium, and negatively charged like anions, phosphate, sulfate, and bicarbonate. Together with these ions help regulate your blood chemistry, maintaining its pH level and proper osmotic pressure, and allowing other tissues to do their jobs, like making muscle contracts and sending action potentials. When measured by weight, the bulk of the solutes in the blood are really plasma proteins. Most of the proteins like albumin and alpha and beta globulins are made by the liver and do things like balance the osmotic pressure between the blood and surrounding tissue and transport lipids and ions. Other run defense for you like the gamma globulin, antibodies that are released by plasma cell during an immune response from fibronegative proteins, which are vital for forming blood clots and stopping bleeding.
Hello everyone, my name is Nur Izzati. So now I'm going to explain about the Buffy coat. So here we can see that Buffy coat is a thin whitish layer that forms between the packed red blood cells and the plasma when a blood sample is centrifuged. It primarily consists of white blood cells, which are leukocytes, and platelets, also known as thrombocytes. For the leukocytes, they can be divided into two types, which are granulocytes and agranulocytes. These two types differ in the presence or absence of granules, as we can see in the picture above. So, for the granulocytes, which are neutrophils, are the most abundant type of white blood cell, which comprising 50 to 70% of all leukocytes. It is very important in inflammatory responses. For eosinophils, it consists of 1 to 4% of the white blood cell, which is involved in the response to parasitic infections and allergic reactions. Meanwhile, basophils make up 0.5% of the white blood cells and release substances such as histamine during allergic reactions. Agranulocytes, on the other hand, can be monocytes, which consist of 2 to 6% of the white blood cells and become macrophages that can eat up intruders. Or they can also be lymphocytes, which consist of 25% to 33% of the white blood cell, in which the B cell produces antibodies, while the T cell fights viruses and tumors. Leukocytes are produced by leukopoiesis, which is stimulated by certain chemical messengers. Platelets are fragments of large cells called megakaryocytes. These fragments are essential during blood clotting, which happens when blood vessels are damaged, as platelets can plug up any holes or tears to seal things off. They flow into the bloodstream in a an inactive state unless needed, dying every 10 days or so and constantly regenerating. Megakaryocytes are formed due to the repeated mitotic cycles that do not perform cytokinesis. So the result is one huge cell with a multilobe nucleus. This then presses against a sinusoid and its extensions burst to release the platelets, which is thrombocytes. These platelets are important during hemostasis, which is the process by which the body will stop bleeding through vascular spasm, platelet plaque formation, and the coagulation or blood clotting.
When you get a cut, this causes damage to the walls of the blood vessels in that area. As a result, blood will escape through the damaged vessels, and that's why you experience as bleeding. But after a while, the bleeding stops. The stopping of the flow of blood is called hemostasis. Hemostasis is the physiological process that prevents and stops bleeding. Hemostasis can be divided into three main phases: vascular spasm, platelet plaque formation, and coagulation. So, vascular spasm. This is also called vasoconstriction. If we look at the structure of the walls of your blood vessels, we see that the middle layer contains smooth muscles. Anytime you see muscle, you know that contraction can take place. In this case, in response to the damage of wall of the blood vessels, the smooth muscle layers will start contracting. This is called vascular spasm. When there's a cut, there are a pain receptor that release chemicals in that local area. Also, the cells that are lining the inner layer of the blood vessel wall also release chemicals. Those chemicals will cause the smooth muscle to contract, and that action will help to restrict the flow of blood. Then, there's the formation of the platelet plaque. Essentially, we have these platelets that are in blood that freely travel through the blood vessels for a moment like this. Well, when there's damage to the blood vessels, it exposes the underlying connective tissue and collagen fibers that's normally in the walls of the blood vessels. As these platelets are passing by minding their own business, they get activated and change their shape. They also will release granules, and the result is they become spiked and sticky. So they clump together around the damaged tissue to form what's called the platelet plaque. This entire process is helped by a glycoprotein that's found in the blood called von Willebrand factor. It helps to stabilize the platelet plaque. This is like a temporary internal band-aid helping to stop the flow of blood until we can have something more permanent. And what's also cool is that as the plaque forms, the platelets also release a few other substances that help the process even more. They release ADP, or we call it adenosine diphosphate. This helps to attract even more platelets to the site of injury. They also release serotonin, which has various functions in this situation. It stimulates and helps to maintain the vasoconstriction we spoke about earlier. Some other substances that are released are prostaglandins and phospholipids. They help to maintain vasoconstriction as well, but also help to activate other clotting factors. For the third and final process, we have coagulation, also known as clotting. We are forming a blood clotting. Now, this is a process that involves a complex cascade of events that involves a bunch of clotting factors. The goal of this process is to form a fibrin mesh that holds the platelet plaque in place so that healing can take place as efficiently as possible. In that cascade, there are two pathways. The extrinsic pathway happens as a result of the damage to the extravascular cells, those not a part of the blood vessels. They are outside blood vessels, hence the name extrinsic. The intrinsic pathway gets activated as a result of the damage to the wall of the blood vessels. Hence, the name intrinsic. It's a pathway that's within the blood vessels. The main goal is that in the common pathway, there's an inactive enzyme called prothrombin that gets converted to the active enzyme called thrombin. Once this thrombin is active, it converts fibrinogen, which is soluble and inactive, into fibrin, which is insoluble and active. Those fibrin strands will form that fibrin mesh that we spoke about earlier that holds everything in place so that healing can happen.
Hi, I'm Nur Amalin in this American from Dietetics course. We'll explain to you about the anatomy of red blood cells. Red blood cells or erythrocytes are a biconcave disk with large surface area and it contains long nucleus or organelles like hemoglobin. Hemoglobin is a large molecule that is made up of four protein molecules called globin. The four globin are alpha 1, alpha 2, beta 1, and beta 2. These globin have a red pigment molecule bound to it that is called heme, and each of these heme molecules have one ion of iron. Each ion molecule can hold on to one oxygen molecule, which means one hemoglobin can hold up to four oxygen molecules. Another thing that you need to know about blood is what type of blood do you have, A, B, AB, or O. These different types do all the job well equally, but they have different flavors for the immune system. All the cells in the body have a plasma membrane with specialized glycoprotein markers on them that act like label or name tags. These markers are the antigen, and our body immune system is totally binding with particular antigen. But if it detects antigen from someone else's cells, including virus or bacteria, then it will send antibody to bind those markers for destruction by the immune system. The red blood cell has specialized antigens on them called agglutinogens that activate antibody to cause coagulation or clumping of blood. The agglutinogens are the ones that define our blood type. There are two types of agglutinogens, which is A and B, and our blood can have either both or neither any of these agglutinogens. So our blood type depends on what antigen we have, such as A type has A antigen, B type has B antigen, AB type has both, and O type has neither antigen. AB type are called universal recipients because they have both antigen and therefore no antibodies for either. Meanwhile, O type is universal donor because they have both antibodies as all blood type can receive the O blood type.
Do you guys know what is the function of the red blood cells? Transporting oxygen is a vital role of the red blood cells. When we breathe in, the millions of air sacs in the lungs fill with fresh oxygenated air. The oxygen then moves into the blood by passing first through the very thin walls of the air sacs and then into the capillaries, which are tiny blood vessels in a network within the lungs. Red blood cells squeeze through narrow capillaries in single file. Hemoglobin molecules inside red blood cells pick up and carry the oxygen, and the cells that are rich in oxygen travel in the blood vessel from the lungs to the left side of the heart. The blood is then pumped around the body. Another function of red blood cell is transporting carbon dioxide. Majority of carbon dioxide is transported as part of bicarbonate system. Carbon dioxide diffuses into red blood cells. Inside, carbon dioxide is converted into carbonic acid by carbonic anhydrase, which is hydrolyzed into bicarbonate and H plus ion. H plus ion binds to hemoglobin in red blood cells, and bicarbonate is transported out of red blood cells in exchange for a chloride ion. This is called the chloride shift. The bicarbonate leaves the red blood cells and enters the blood plasma. In the lungs, bicarbonate is transported back into the red blood cells in exchange for chloride. The H plus dissociates from hemoglobin and combines with bicarbonate to form carbonic acid with the help of carbonic anhydrase, which further catalyzes the reaction to convert carbonic acid back into carbon dioxide and water. The carbon dioxide is then expelled from the lungs.
Hi everyone, I'm Penny,
and I'm going to continue with the blood disease. And today, we are going to focus on Rhesus disease. So, what is Rhesus disease? The Rhesus disease begins with the blood. Every one of us can be divided into one of 4 major blood groups, including blood group A, B, O, and also AB. However, there is another category that can differentiate us, the RH. So what is the RH or RH factors? RH factors is actually a certain types of protein that found outside the red blood cell. So people who have the protein are considered as RH positive, while people who are missing this RH protein are RH negative. The majority of people, which is about 85% of them, are RH positive, while the rest are RH negative. And these 2 blood groups are incompatible. That means if they mix, deadly reactions can occur. So RH incompatibility can occur when a RH negative woman has a baby with a RH positive man. So when this happens, the baby will have 50-50% chance of having either RH positive blood type or RH negative blood type. It is very dangerous when a RH negative woman becomes pregnant with a RH positive baby. If the baby's red blood cell get into the mother's bloodstream, which can occur during childbirth, miscarriage, or abortions, the mother immune system will consider them as foreign invaders and then will create the antibody to destroy them. So women that are experiencing their first pregnancy usually don't have to worry about RH incompatibility because it takes longer than 9 months to develop the antibodies. And these immune reactions will begin during a mother's first pregnancy, but is only complete after the baby is born. That is why the first babies are typically spared from RH disease. And this immune system built during the first pregnancy will never forget and will leave all the future RH positive baby at the risk of getting RH disease. RH incompatibility becomes a problem if a woman becomes pregnant at the second time and having another RH positive fetus. These antibodies will put the second fetus at risk. Once the body realizes the fetus is RH positive, the antibody can cross the placenta into the fetus blood and attacks its red blood cell. And whenever this happens, these red blood cells are going to hemolyze, which will reduce the count of red blood cells in the baby's body. So, let us recap back what do the red blood cells do actually. They help to transport oxygen throughout the body. But in this case, that will not happen because the antibodies are attacking the red blood cell and decreasing the number of red blood cells. This will cause the several animals to happen and jaundice as well. These antibodies will also attack the fetus with terrible consequences like permanent brain damage, heart failure, or even death in the unborn or the newborn baby.